photos as you wish during my presentation of my slides. In 2020 approximately, I remember very vividly listening to a radio program where somebody was talking about nanobiology and how important it was and how interesting it was, and I was thinking, how do you study nanobiology? And I decided then and there I will not study nanobiology. Here I am, 25 years later, actually discussing exactly that. So my name is Lötvall. I am actually a clinical professor, but I've done mostly translational research. I have a number of disclosures to list there as well. So my presentation will be divided in principle in 4 different sections, and I'll talk a little bit about the early discoveries and how we entered the field of extracellular vesicle research, the initiation of ISEV, and how that was absolutely crucial for the development of new methods and simplifying the ways we do research. I did a detour. I was still employed at the University of Gothenburg part-time, but I did a detour for a few years as chief scientist at this startup company called Kodiak Biosciences in Boston. And that was a very interesting experience. I'll touch on that slightly. And then I'll talk about my own drug development ambitions currently and how we are pursuing those. One thing that is, you have a number of company logos up there. What is extremely important is that we are doing all this discovery and development at an academic level at the University of Gothenburg through different types of funds. In Sweden, the professors or the teachers own their patents and can translate them to commercialization or to proper development, which is unusual. So we came into this field via this paper in Nature Cell Biology. Many of you have read it, many of you have cited it, so thank you very much for that. And it was an interesting journey. We were inspired to study what was called exosome then by Esbjörn Telemo, who is an immunologist, now retired at University of Gothenburg. And he was looking at tolerance factors, and we were interested in how the lungs and the bone marrow communicate to induce inflammation. So basically we had a hypothesis, and that is that exosomes from the lung go out into the blood, enter the bone marrow, and there get the eosinophil inflammatory cell production happening. And Anna-Karin was doing those experiments quite extensively for a number of months, and we had some good data and some bad data, and we did not know how to deal with this. Until we had a lab meeting, we went through it all, and then Karin Ekström, who was my PhD student at the time, suggested, why don't we go in vitro and look at these exosomes from mast cells then being allergy associated. And we did. We isolated the exosomes. We did proteomics. We found more than 150 proteins at a time when there were only 32 proteins described in vesicles. This is around 2004, 2005. And many of those proteins were RNA associated. So if they're RNA associated, maybe there's RNA in these vesicles as well. And we asked that question at that meeting within 10 minutes of looking at that data. And then we sort of followed the data and asked ourselves, is there RNA, instead of trying to push an allergy question into that. So this is a matter of follow the freaking data. Remember that. That's an important thing to remember. And only a few weeks after we asked the question, we could see small RNAs, especially in these smaller extracellular vesicles, which was different, clearly different than the RNA from the cells. And we could see that this RNA was functional. We found at least some functional mRNAs in these isolates that could translate to proteins in vitro. So this is how we started the field, and it was an important platform. But I'm not really an RNA person. I'm more of a doctor or trying to understand. And I think one of the things we wrote in that article, which we should remember, is that we were thinking maybe these extracellular vesicles, these exosomes that they were called at the time, could be used as therapeutics. And we were discussing gene therapy at the time, but that's certainly one possibility. but this dream of developing a therapeutic using this technology is something I have carried with me throughout this period. And then comes the period of methods. So because, of course, that paper we published in 2007 was not perfect from an isolation perspective and characterization perspective. It was reasonable. It was, at the time, it was a state of the art of the time, but there could be improvements certainly happening. And there was a lot of methods, efforts being happening during this period between 2007, I would say, until around 2014, some 15 maybe even. And I had Cecilia and Maria and Ganesh and Rosella pushing for these method developments and a number of other students. But most importantly, I got to know Yongsong Go, who's here in the front row actually. And he became a little bit of a mentor and a teacher for all of us and helped us push the thinking further. So without Yongsong, I would probably not be standing here today. So thank you, Yongsong. So we have a number of different papers that were published in that period, and clearly it was not papers that would be easily published in any biology or medicine journal, but they were difficult to find a platform to communicate that science. So we felt that we had to start the journal for extracellular vesicles, and we did that on the 16th of April 2012, the first day we had the formal ISEV meeting. And there were a number of different papers that were published that got a lot of citations, even though the journal, of course, did not have an impact factor until 2019. So in the first issue, we had 8 publications from a number of prominent researchers, and that was the first day of the formal ISEV meeting. ISEV had been conceived at the meeting that Clotilde Terry and Graça Raposo arranged in Paris the year before in 2011. And this was a closing ceremony of that meeting. We had 500 participants. Nobody wanted to go home. Everybody had learned something from others at this meeting and interacted, and was very happy to find people asking the same questions, struggling with the same methodologies. So that was quite interesting, but also, like here, we had a lot of fun as well, and you'll be getting those pictures at the end of my presentation. So then we come into this period around 2015, 16, 17 maybe, when I was not the only one wanting to develop extracellular vesicles as therapeutics, but there were a number of different scientists around the world that started companies in parallel. And some of these companies are still around, some of them are perhaps not as strong as the others. So certainly there are a number of people out there. So I went to Kodiak in early 2016 and helped them establish methods to isolate extracellular vesicles and to characterize them, etc., because basically they did not have that technology at hand at this time. So they raised a lot of money and they performed a lot of work. But there was one decision, I think, that was maybe not super good, and they wanted to use the extracellular vesicles, the exosomes, as drug delivery vectors. So the CEO was feeling comfortable with developing small drugs, and putting a small drug into the extracellular vesicles was something that was natural to him. And one of the things that was the first one, actually after I left, this started taking off, was loading a STING agonist, an immune stimulator, into HEC293 exosomes. And Suchul Jang, actually I recruited him from my lab in Gothenburg, and he was also the student of Yongsong before that, came over and did a very good characterization of these vesicles, showed that they had therapeutic potential if they were loaded with STING agonist. And then there was, the thinking was that exosomes enabled the drug. It was not the other way around. It was not the drug enabling the extracellular vesicles, or there was no synergy discussion at this stage. And then there was a clinical trial that was performed that was, what should I say? It was maybe not optimal, it was suboptimal. It was 15 different types of tumors, direct tumor injection, and there were 23 individuals. So no clear direction in that study, and it of course came out negative. There were some patients, some cases that seemed to have some beneficial effect. There were other drugs that were also being developed. Exosomes was also tested and had some preliminary interesting data in 2 patients, I think, but otherwise they did not pursue any more clinical trials. And unfortunately, Kodiak closed in early 2023. And I want to emphasize again, 5 years after I left the company. My mindset when we now are developing extracellular vesicle therapeutics, that is to, again, follow the data, FFD, and that is try to harness what the extracellular vesicles do. Don't try to force them to do something that is unnatural to them. Try to amplify what the extracellular vesicles do. Try to make them do it better than they do it themselves. And then we have to document safety, and we can all do all of this within an academic setting at a university level. But when it comes to advanced toxicology and preparing GMP material and so on, very few academic settings, Eva and Mario is an exception, can do that. So usually you need to step out and go into a company situation. So what do the EVs do then? Well, from my perspective, with a background in allergy, in immunology, it is absolutely clear that vesicles are taken up by immune cells. And the most professional cells taking up extracellular vesicles are dendritic cells and macrophages. And if you look at most studies and compare different types of cells, those are the cells that really take them up and process them. So those are good target cells, and you can target them for different purposes, but of course to influence and regulate the immune system. So throughout this is part of the presentation where I go through our internal kind of drug development plans, academic drug development. We have several generations, several stages and improvements in the technology that we think are important to make them, to optimize the therapeutic potential of these extracellular vesicles. And there are 2 platforms we're working with. One is bacteria. We make vesicles. We don't take the natural vesicles from the bacteria. We produce vesicles. I'll explain why in a little second. Or we use mesenchymal stem cell or cells that are supposed to behave like mesenchymal stem cells. And of course, these do absolutely opposite things. Like the bacteria, they stimulate the immune system, activate the immune system, whereas the macrophages or the mesenchymal stem cells, they attenuate or slow down or block the immune system. But it is the same target cells in principle. So we started with bacterial vesicles, and there are 2 reasons for that. Of course, we were inspired. Kyung-soo, also recruited from Yongsong's laboratory in South Korea, had experience working with bacterial vesicles. We were also inspired by the work of Yongsong, published just recently, and there are even vaccines available with bacterial vesicles that have been approved for clinical use. But there was one more reason that I want to warn you all a little bit about, and that is when I had left Kodiak, this was 2018, I had a non-compete agreement with them. So I could not go in and study or do competitive research on eukaryotic vesicles, HEC293 or mesenchymal stem cells. So that was something I avoided, and that's another reason. So when you're at an academic setting, try not to have a non-compete agreement, even if you've worked with companies before. So what Kyung-soo has done is to isolate vesicles from bacterial isolate membranes from bacteria and then made vesicles from those membranes. And basically, it's open membranes that he produces, and then those membranes can close and become vesicles with certain procedures. And this is what happens then is you remove RNA and you remove DNA from these structures. And they look, as you can see on this electron microscopy picture, just like any extracellular vesicles with this donut shape. So the interesting thing with these vesicles, and I think that's really the what we call inventive step, is that surprisingly, they don't induce inflammation in mouse macrophages. They don't induce cytokine release from mouse macrophages. But when you put them on dendritic cells, they activate the dendritic cells very efficiently. And specifically then interleukin-12 and not IL-4, which of course is important for immune oncology purposes or immune stimulatory purposes. So you have two opposite functions here in two different cells with the same vesicle. So safety is seemingly then important, and can these be given in vivo without causing side effects? And in fact, we did inject these. This is intraperitoneal injection in the mice, where these semi-synthetic bacterial vesicles did not cause cytokine release or fever or systemic inflammation in these mice. And why is that? Well, remember, these vesicles are empty. They're reduced cytosolic proteins. There are no DNA or almost no RNA in them. And if you put them on an assay looking at which TLR receptors are activated, is totally different. So the green one here is the normally released outer membrane vesicles from the bacteria. They activate all of these TLRs, whereas the semi-synthetic bacterial vesicles activate TLR2 and TLR4 only, and there's some activation of the other ones. And clearly, so you have reduced RNA, you have reduced DNA, and those TLRs that are activated through those pathways are not activated or much less activated with these vesicles. And that's probably an explanation why they are they are much safer. But clearly, the LPS is there, and the LAL assay, if you run a LAL assay, you will find the LPS there. So, but still, they are safe in these systems. And can they work as vaccines then? And we tested them as bacterial vaccines in both E. coli or Pseudomonas aeruginosa models, and we published that a few years ago. I'm not going to go into that in detail, but we do have evidence that yes, you can actually inject these safer bacterial vesicles, and you get an immune response against these bacteria, and you have a protection against infection to those bacteria. We also put the COVID spike protein on the vesicles and could get an immune response against the virus as well, antibodies against the virus. But this is not work we are really pushing right now. We're more using this for immune oncology purposes. So in principle, these features of the bacterial vesicles can be very useful for immuno-oncology purposes. So we use these empty vesicles, and then we mix it with another technology that was developed by Rossella Crescitelli over the last 10 years, again, inspired by Yong-Song Go and in this process. And that is to try to isolate vesicles from tissues. And she's been spending almost 10 years on that, I think, and has a paper in Nature Protocols a few years ago now, where the methods are very clearly described. And if you look at these electron microscopy pictures, the negative stain on the top left, which is from ovarian cancer, or the lower one, which I believe is from melanoma, is a cryo-EM picture. You see they look very much like vesicles. And if you characterize them, they have all the features of extracellular vesicles. What we're just working on as well is that isolating vesicles from frozen tissues versus fresh tissues. And actually, my bias was that you have to have fresh tissues. But it seems that using frozen tissues is just as effective. You get very, very similar extracellular vesicles when you isolate the vesicles from the frozen tissues. And that's something that Rosella is pursuing as a manuscript. And why we would using these tumor tissue vesicles? Well, the reason is that they are full of mutations. The cancer mutations are there. They're sort of enriched on the surface of the extracellular vesicles, and these vesicles are, as I mentioned, very easily taken up by dendritic cells, for example. Now, if you just inject the vesicles themselves, you don't get very much of an immune response. You need to boost the immune response somehow, and that's why we mix them with the synthetic bacterial vesicles, and we've tried and compared with other adjuvants, but the synthetic bacterial vesicles are more efficient than even Freund's incomplete adjuvants. So, but the concept here is the mutations are on the tumor tissue extracellular vesicles. So here we have two examples. One is colon cancer to the left and melanoma to the right, and you can see here that the bacterial vesicles. Let's focus on the melanoma one. If you look at the red curve, that is bacterial vesicles alone, and that inhibits, slows the tumor growth. And the yellow one is bacterial vesicles plus a checkpoint inhibitor treatment. This tumor, B16, is not sensitive to checkpoint inhibitors, but if you combine it with something that makes the tumor hot, in this case it's synthetic bacterial vesicles, you get this synergistic anti-tumor effect. And also this immunotherapy can attenuate or slow or reduce the metastasis process. And again, these are synergistic findings, so they're kind of, from that perspective, surprising. We've continued and tried different approaches. Obviously, the vesicles are in the tumor tissue. Can you then inject the bacterial vesicles directly into the tumor tissue? And there they engage with the dendritic cells and the existing extracellular vesicles in the tissue that have the mutations. And clearly, you can also get inhibition of the tumor growth by doing this. This is in melanoma. So, and if you look at the mechanisms of how these treatments work, it's clearly an activation of adaptive immunity, and you get antibodies against the tumor, and you get T cells against the tumors, against the cancer. And this process is at least partly happening within the local lymph nodes, distant from the tumor site. So we have then the last generation that we have so far with extracellular vesicles, and we have the synthetic bacterial vesicles that have reduced TLR activation, and then we still put another innate immunity stimulator into these vesicles, which is a STING agonist. They don't activate TLRs, seemingly, but they activate other parts of the innate immunity system. And when we do that, if you look at the left side of the curve, in the yellow column, you can see a massive enhanced stimulation of dendritic cells when these bacterial vesicles are loaded with the STING agonist compared to the bacterial vesicles alone. And also in the colon cancer, this is intratumor injections with this STING agonist, you can see total inhibition of the tumor growth, and that was not possible with the previous approach when we mix the extracellular vesicles from tumor with the bacterial vesicles. But in this case with STING agonist, yes, it is possible. So within this bacterial vesicle therapeutic development, we have multiple generations. We have the first one that we show that they're non-toxic. They can be used as vaccines against bacteria. We can mix these vesicles with vesicles from the tumor tissues, and by mixing them we can get an immune response against the tumor. We can also inject them into the tumor and again get an inhibition of the tumor growth using that approach. And we do all of that with an improved safety. And then we have this last step where we get even further enhanced immune responses if we have loaded a STING agonist into the bacterial vesicles. And that worked very well in the colon cancer. And I should emphasize here that this generation 3 did not increase the risk or the safety, reduce the safety in any way. The systemic effects were not enhanced in any shape or form. And clearly, the conclusion is that this happens through a adaptive immunity stimulation of the immune system in these mice. So we have another approach which is the same, but the opposite at the same time. So we're trying to develop anti-inflammatory treatments. And having a background in lung inflammation and asthma and things like that, you want to attenuate the inflammatory response. And mesenchymal stem cells have been around for a long time and been tested in these systems and have shown quite good anti-inflammatory effects. And then mesenchymal stem cell extracellular vesicles come, and I'll show you our first slide, our first study in my next slide. But then there are possibilities of moving even further. So again, the target is these dendritic cells or the macrophages. They are the ones that take up these vesicles. And if you want to stop a severe inflammation, it's probably macrophages that dominate. If you want to attenuate something like an autoimmune disease, perhaps it is the dendritic cells that are the target. And this is the first study that we were part of, which was with Milad Riasfar, who is in the room, and many of you have met him. We worked around 2015 on these things. It was published in 2019. With this neurodegenerative model, we can see around a 50% inhibition of the clinical picture in these mice by, in this case, a single injection of mesenchymal stem cell extracellular vesicles. So after we did this project, we expanded our interest in mesenchymal stem cell vesicles and have continued working on these. And we should also remember that there is clinical translation ongoing today. There are two double-blind placebo-controlled studies that have been published. One is from Direct Biologics, a US group that published in Chest 2 years ago now, that mesenchymal stem cell extracellular vesicle infusion could improve the clinical picture of COVID lung injury. And similarly, this is an academic group from Iran, published in JEV, could show that, again, confirm that there was an improvement in the clinical picture and more rapid recovery from the disease if they were given intravenous mesenchymal stem cell extracellular vesicles. So this is obviously something to remember. There's something happening, but perhaps these effects are not sufficient to really hit that home run with these drugs, and I usually suggest that it's really probably good if we can enhance this effect even further. So we've been discussing and thinking about how to make these mesenchymal stem cell vesicles more powerful, more potent, more effective for a number of years now with Kyung-Soo and others in the lab. And one of the fundamental things that we're doing is to load them with appropriate cargo molecules. And you can think about many different cargos. We've been working a lot with peptides. We have a favorite peptide I will come back to. But this is one way of doing it. And try to load something that adds value to the mesenchymal stem cell vesicle. That's the important aspect. And again, we use a very similar approach to what we've done with bacteria. So in this case, we can make vesicles from the membrane or take the vesicles themselves that are released. We process them through ionic shock and make these open vesicles that empties the DNA, empties the RNA. And then we can load them. In that process, we can load them, and then they spontaneously close, or you add energy and they close. And by doing that, this is what they look like after the whole process. They look just like extracellular vesicles, even though they do not have DNA, or very little DNA and very little RNA inside, and reduction of cytosolic proteins. And that means they're empty, and that means we can load them with a lot of different stuff. Now, do they have biological activity when we remove things? Yes, they do. You can see here in the yellow, these are the empty vesicles. They have the same anti-inflammatory effects, and this is the mouse macrophage cell line that we've studied. You can see the dose-related inhibition of cytokine release by these empty vesicles. So the multiple conclusions from this. One is the membrane is very important. The other is maybe the RNA and the cargo proteins are not so important for these type of assays that we are running. So you can make empty vesicles and they still have function. We've tested these in multiple different animal models. This is just examples of asthma model or LPS exposure. They are always still anti-inflammatory also in the in vivo situation. Now, we focus quite a lot on inflammation, severe inflammation that is induced by bacterial outer membrane vesicles. That's in principle LPS. That's how LPS is present in nature. And all these TLR, these outer membrane vesicles, they activate the immune system through TLR receptors, as I discussed before. And there's a common intracellular messenger for most of those, all except one, and that's MyD88, which is a very difficult molecule to target. But we have found a peptide that is actually quite efficient in blocking this molecule, and we've loaded the vesicles with that, and you can get thousands and thousands of these vesicles inside of each vesicle. So when we do that, and this is, we see a dose-related inhibition. I'm not showing that data, but you see here in vitro, the peptide has some anti-inflammatory effect. You see the blue column, and the red column is the vesicles around 50%, like we see in most. These are fairly high doses. But if you have the vesicles loaded with the peptide, and that's the same quantity of peptide, the same number of vesicles as in the previous two columns, you see a much more enhanced anti-inflammatory effect. And you're coming up to the 80-90% inhibition of inflammation, which is probably what you need to make a really kind of home run anti-inflammatory drug. So these are synergistic effects. Same thing with in vivo, and this is, the peptide is absolutely inactive when it's given in vivo. But if you have the peptide loaded into the mesenchymal stem cell synthetic vesicles, you see the synergistic effect, and again, right, 85% inhibition of inflammation. So that means that the peptide is working. If it's in the vesicle, it's enhancing the vesicle, and the vesicle is enhancing the peptide. And that's basically what's called synergy, and that's what I believe is necessary to successfully develop a really potent, efficient drug to treat severe diseases. Marcus is also here in the room somewhere, my PhD student. He's been working on developing targeting moieties on vesicles to be able to get to specific cells, and this is the beginning of a quite big effort, I think, in developing really cell-targeting extracellular vesicles. In this case, he's using HEC293 cells, and he's expressed LFA1, that's CD18, and CD11A. It's not easy to overexpress, it's natural material, to target ICAM1, to specifically try to target endothelial cells. And here he can show that the LFA1 vesicles bind and are taken up efficiently by endothelial cells if the vesicles have the LFA1 on their surface. And likewise, if you block ICAM1, you can see a dose-related inhibition with the neutralizing antibody of the uptake of the vesicles in endothelial cells. Now, of course, we loaded these vesicles with our peptides to try to block inflammation. In this case, we're looking at IL-8 release. And you can see the yellow line is what the peptide does alone. The vesicles alone do nothing, but if you deliver the vesicles with these LFA1 expressing vesicles, then you get a much more enhanced anti-inflammatory effect. Again, what we call synergy. There's one more clinical translation that is ongoing that I think most of us and many of us are aware of. That's being developed by a group from Israel, Nadir and colleagues are developing a HEC293-derived vesicles expressing CD24. And that is an anti-inflammatory molecule, and they have taken that into the clinic, specifically in COVID-19, and seen clinical benefit. But unfortunately, these studies are not placebo-controlled, and I think they're working very hard on pursuing phase 3 clinical trials in the near future. Of course, we have also put CD24 on HEC293 vesicles, and you see that with the red columns here, and we do see dose-related anti-inflammatory effects of these vesicles. But we also put other molecules that we think are interesting, and you see that in the orange and in the blue columns, we can get anti-inflammatory effects out of HEC293 cells that are otherwise not anti-inflammatory by changing their surface profile. So these generations of extracellular vesicles are, I think, quite promising. You can make vesicles from the cell membrane that creates high yield. You can load them with different molecules such as peptides, or we also have data on different drugs that you can load into the vesicles very efficiently. And they have this fourth generation where you can put either homing proteins on the surface of the vesicles. You can probably combine the homing with the anti-inflammatory moieties as well to get a more potent and a little bit targeting extracellular vesicle therapeutic. So personally, I think this is a very, very exciting field. And if you look historically back and look at monoclonal antibodies, they were discovered in 1975, that's 50 years ago, and we're still new drugs coming out based on monoclonal antibodies, and I think that's really, really interesting. I believe that the field of extracellular vesicles will see the same kind of growth if we just get over that little hurdle of getting the drug registered in the beginning. So I see these drugs as this technology as very promising, and I think we can really change clinical medicine if we just use the right cells for the right purpose and follow the freaking data. And with that, I want to thank you for your attention, and I want to thank everybody that I've been working with over the last 20 years. Thank you.
Thank you, Jan, for the brilliant overview we received yet now in your talk. It was an example of how you really always
Please put the slides up again. Please put the slides up again, please. Thank you.
Thank you for interrupting. How you brought all the ideas into the field and how we should really discuss the concepts. So one issue I really have to challenge you immediately. You always say we need a stronger effect, we need a high potency, but biology is not black and white. So if it is a decent effect, it might be maybe even better for our body if we try to treat
Yes, I always say that, don't I?
Huh?
I always say that we need to get better effects, stronger effects.
And stronger, but I would see what is the impact on the body if you have a black and white. So we very often see animal data where we have black and white, and you cannot repeat it in the human systems.
You know, I don't exactly know how to, as a doctor, you're a doctor, I'm a doctor, you're also a doctor, right? Medical doctor. So we want to reverse a disease process, right? We don't want to improve it a little bit, right? And that's my mindset, and that's why I push to make our vesicles as potent and as effective as possible, right? So potency is also helpful because potency means how much vesicles you need, and if you put something beneficially in there, maybe you need 100 times less or 1,000 times less. That's reduced cost of goods. I'm sure that's something you think about as well. So I think there's more than just efficacy, but also potency. By the way, you can if you go with that link, you can get a lot of historic pictures from ISEV previous years.
Yeah, thank you very much. And if I may add, sure, the potency, we need to compare the effect with the existing drugs, which is, I don't think has been done so much. And also drug resistance, and see if the mechanism could overcome many of the resistances we have to drugs. But having said that, maybe we also open the discussion if there are questions from the audience. We heard very provocative
Please proceed to the mics.
Okay.
Lisa is there.
I'll ask a question.
Please.
So, thank you for that fascinating talk, Jan. I have just a simple question. It looked to me like your bacterial empty EVs were kind of like a different kind of adjuvant. So could you comment a little bit about how the bacterial EVs as an adjuvant compare to current adjuvants that we use for vaccines?
Yeah, so we have done direct comparisons with Frontine Complete and alum and so on, and we see higher efficacy with this. That doesn't mean that we've done a whole detailed comparison for safety and efficacy, because we're not really pushing for that. But yes, you can use these bacterial vesicles as adjuvants for sure.
Maya?
Hello, thank you, Jan, for a beautiful overview of, I'm here. So my question is about actually your opinion about the perspective. This field yielded a lot of great results and a lot of great promises. However, it's not still progressed into the clinic. Do you think that we have to be back to basis and to first discover mechanisms of action so we can harness this data to produce what would be most efficient in clinic and able to be upscale in production? Because I think a lot of these approaches which are actually, let's try something, and if it works, let's then think about how to upscale it or to see how it works. Maybe, what do you think? Is it important to come back to basis and first discover mechanisms of action of what naturally happens?
So I think we're extremely close to clinical translation, and I'm not ready to say that clinical translation has failed. I don't think it has failed. These clinical trials that I showed you do have p-values that are significant. I am hoping that we can find drugs, EV-based drugs, that are more effective and maybe more potent as well. And that's where I'm pushing my thinking, right? But it doesn't mean that there are other things happening. I mean, there is people selling extracellular vesicles quite successfully. In the cosmeceutical field, and some of those products are promising, and some of them are maybe not so promising exactly. But clearly there are clinicians in that field that find some of those products being promising. Still, they're not injected necessarily or proper disease targeting drugs at that stage, right? For that, we have this system that we have to go through, which is regulators require us to do toxicology, to do appropriate clinical trials with appropriate endpoints and so on. And I think some of the failures have been the design of studies, maybe the design of clinical studies, so one of the clinical trials that I mentioned was huge, more than 100 people, but still barely significant, right? And maybe there were design issues around that. I don't know the exact design of that trial, and not everything is reported either. So I think with the right designs, I think we will get drugs approved. And I believe the CD24 should get approved at least when they have a phase 3 double-blind placebo-controlled study. That doesn't mean that the big pharma will take it and sell it. That's another issue, right? That's another hurdle you have to find. But I still have hope. And mesenchymal stem cell EVs, the standard ones, that maybe are not as potent, will still get approved. I believe they will get approved.
More likely than the engineered vesicles? More likely than the engineered vesicles, or so?
No, it's a chain of events, right? It will take a time before the engineered vesicles will be approved, have gone through toxicology testing, and so on. I have strong beliefs, and there are many people in the room that are running, genetic engineering of cells to make amazing vesicles with unique features. And that's very promising, but it will take more of safety and more of a regulatory process to get those approved than the mesenchymal stem cells, which have been tested for so many years in so many studies, both as cells and now as vesicles.
Thank you.
Okay, let's give the chance to some other questions, brief questions, and brief answers.
Short answers, please.
Yes, ma'am.
Okay, I have a question, and basically about your comments about the EV membrane protein is more important. The cargo is not as important, because I saw the field people spend a lot of time to do the proteomics, the microRNA sequencing, try to look at the cargo. So actually, I just want to say your insight about this. Do we still need to do that?
Can you repeat the question?
We acoustically did not get your message.
Proteomics in the
Could you repeat?
It's difficult to hear from up here, actually. Proteomics of the vesicles?
My question about your comments in the slides, you talk about the EV membrane is more important than their cargo.
Yes.
So I think in the field, people spend a lot of time trying to do the microRNA sequencing, do the proteomics,
Yes.
to understand the cargo. But look like we do not have to do it. We will have to focus on the membrane protein.
So if you look at proteomics of an extracellular vesicle isolate, you get usually about 700, if you have a proper approach, 700 membrane proteins. And those and the lipids and what else is on the membrane seem to be the most important part for these type of assays. There may be other things that the RNA and the cargo can be important for.
Okay, thank you.
Final question, please.
Okay, my question is very similar to the question previously asked. In your opinion, well, first of all, thank you for a beautiful overview of your work. My question is, in your opinion, do you think that protein will be more important in terms of cargo versus RNA?
And if I might add, are you sure that when you open the vesicle, the cargo will not stick on the surface?
Yeah, I mean, we cannot isolate RNA from the vesicles, right? So, you're coming back to the corona? She likes the corona. So clearly, for these assays, and I repeat, for these type of assays that we run in our labs, which needs to, you stimulate something and you need to harvest your supernatant 24 hours later. For those assays, in our hands, definitely the membranes or the membrane components are much more important than the cargo components. That doesn't mean that in chronic diseases or in longer exposures that the RNA may still be important. But for these rapid changes in our hands, the RNA is not as important. I stand by that very strongly, actually.